The overall goal of this procedure is to prepare sequencing libraries of RNA viruses directly from clinical samples to enable viral surveillance and evolutionary studies.
This method can answer key questions in the fields of viral genomics and evolution, such as how RNA viruses mutate within and between hosts, and how they spread within a population. The main advantage of this technique is that it's unbiased. It requires no prior knowledge of viral sequence for library preparation and can be used to sequence any virus.
The implications of this technique extend towards detecting and combating viral diseases like Lassa fever, because the diagnostics and the therapeutics we develop rely fundamentally on the genome sequence of the virus.
Demonstrating selective depletion will be Adrianne Gladden. Demonstrating cDNA synthesis will be Winnicki, and demonstrating DNA library construction will be Dolo, Nosamiefan.
To begin, prepare 5x hybridization and 10x RNase H reaction buffers and nuclease-free water with linear acrylamide carrier as described in Table 1 of the text protocol. In a 96-well PCR plate in a metal block on ice, set up a hybridization reaction by combining previously extracted and DNase-treated viral RNA and ribosomal RNA depletion oligos and oligo-DTs. Note, all reactions in this protocol are prepared and distributed into strip tubes for use with a multi-channel pipette. Vortex the reaction gently and thoroughly, then centrifuge at 280 times G and room temperature for 1 minute.